So ReNeuron is a pure stem cell therapy business. We're harnessing the potential of stem cells as therapeutics, and our focus is on allogeneic cell therapy treatments targeting significant areas of unmet medical need. We have a number of platforms which I'll run through in summary today. We're relatively small as a business, about 65 employees based primarily in the UK, and we also have a small site now in Boston, and we're quoted in London on the AIM market. So the platforms we're working with fall into three types, as you see across the top of the slide here. We have two pure cell therapy assets. One's called HRPC, which stands for human retinal progenitor cells, and CTX is our immortalized neural progenitor cell line. The HRPCs are currently targeting retinitis pigmentosa, and CTX is targeting stroke disability. Both products have been developed or formulated as a cryopreserved product with a long shelf life in both cases over a number of years to build out the CMC processes around that at GMP level. And we have early phase 2 data in both programs as well. Early phase 2A data in the case of RP from a US study that's currently running, and phase 2A data from a UK study we completed last year, and we're currently embarked on a phase 2B study with CTX again in the US. Both of these programs are actually partnered now in China or the Greater China market with Fosun Pharma. We announced that deal a week or so ago. We're also harvesting exosomes from our CTX cells. These are nano-sized vesicles and are starting to come more onto the radar in terms of their potential both as therapeutics in their own right and also as carriers or vectors, novel vectors for delivering third-party therapeutics, which is how we're exploiting our own CTX-derived exosomes. And I'll come back to that towards the end if there's time. And you've got the pipeline chart there. So we're quite focused in terms of our programs at the moment, with RP and stroke disability being the core programs we want to generate meaningful data with across these assets. So looking at the HRPCs in a little bit more detail. So as I mentioned, we're targeting retinitis pigmentosa. So again, this is, if you like, a gene-independent approach to targeting RP, which is, as you know from the previous presentation, is a collection of gene defects, essentially. Ultimately, it leads to blindness and is characterized essentially by loss of peripheral vision, as you see in the images here. Our own program in RP has orphan drug designation here in Europe and also in the US, and also fast track designation from FDA. We did some IND enabling work over a number of years. A lot of this is now published, showing that HRPCs could preserve vision in an RP preclinical model, RCS rat, dystrophic rat. And we saw the ability of our own HRPCs to preserve vision and also the ability of those cells to integrate into host retina and provide trophic support to the host cells. We also did some larger animal work. We generated essentially a pig-derived homologue to the HRPCs, so pig retinal progenitor cells, if you will. This was a non-functional model to show evidence of differentiation and engraftment in a larger eye, and that's exactly what we saw in terms of the ability of these PRCs to differentiate and integrate into host retina. We don't need to, even though these are both allogeneic products, both in RP and also with CTX in stroke, we don't require to immunosuppress patients in any of the clinical work we do. And the cell types and cell lines were selected partly on that basis, their ability or their immune characteristics, if you will. In terms of the clinical work in RP, as I mentioned earlier, we're currently in the middle of a phase 1/2A study across two sites in Boston with Jason Commander and Praveen Dougal as the investigators, both key opinion leaders in this area in the US. We have completed the phase 1 element of this study. It was a dose escalation study, and we've now moved into the phase 2 element using the highest dose that we used in phase 1, which is 1 million HRPCs injected subretinally to the back of the eye. So relatively straightforward retinal surgery to get the cells in. What we announced recently was some very encouraging early phase 2 data. In the first phase 2 cohort that we treated, where all 3 patients that we treated so far showed a very significant and quick improvement in vision, equivalent to 3 lines on the ETDRS chart, which is what you see on the right-hand side of the slide there. Obviously, what we need to see is a durability of response at that level. 3 lines or more on this chart is clinically meaningful in the eyes of certainly the patients and certainly in the eyes of the FDA as well in terms of clinical development. And we're actually going to be presenting a little bit more updated data on this at ARVO in Vancouver at the end of this week. We'll be following these patients up for 12 months. Primarily, this is a safety study at the end of the day, although obviously the phase 2 part of the study is monitoring on a number of efficacy measures as well, both visual acuity and also visual field and so on. If we're successful with RP as the first indication, then clearly there's potential here to broaden out into related indications such as cone rod dystrophy and so on, and that's certainly in our planning. Okay, so just moving on quickly to the CTX cell line. So as I mentioned, this is targeting stroke disability. Again, it's a cryopreserved cell therapy product manufactured under GMP. We have a number of CDMO relationships to do that right now on a long shelf life. And essentially what we're trying to do here, as we are with the HRPCs, is to get these living cell products as close as we can to a standard biopharmaceutical paradigm in terms of ease of manufacture, shipping, storage, and clinical use. So we're targeting stroke disability with our CTX cells, and it's really the consequences of stroke that we're looking for CTX to engender an effect. So in other words, we're targeting patients that have suffered a stroke, survived it, but have a residual motor deficit. And that is where we have the preclinical evidence to show that CTX can ameliorate the effects of those motor deficits in validated preclinical models, and we have some early clinical evidence now as well. So we're treating patients currently at least 6 months after their infarct. So they have stabilized in terms of any spontaneous recovery that most stroke patients, if they survive the initial insult, are going to see. But after 6 months, if you haven't fully recovered, then you're not really going to recover any further. And that's, if you like, the sweet spot for us in terms of treating patients and trying to get them further up that recovery curve. In terms of the mechanism of action, again, this is a paracrine effect, as you'll see with most allogeneic cell therapy approaches. You're looking for those cells to work trophically through release of paracrine factors to stimulate, in this case, angiogenesis, neurogenesis in the brain, and so on. And we do have some preclinical published evidence for those mechanisms as well. So in terms of clinical development with the programs, we've run 2 clinical studies so far, both in the UK. These were both single-arm studies. The first one, Pisces 1, was a safety study in 11 patients and a dose escalation study. And having shown a good safety profile with the cells in that study, we used the highest dose of 20 million cells and took that into our phase 2A study, again in the UK, this time in 23 disabled stroke patients. Again, a single-arm study. The important thing to note here is that we monitored patients for a stable deficit before they got the cells implanted. And the cells are put in through stereotactic surgery. So again, it's a procedure that takes about 90 minutes through a single needle tract directly into the basal ganglia region of the brain. And the patient will typically stay in overnight or maybe 2 nights to monitor for any complication, bleeds, and so on. And then they're back home. And that's been the case for all of the patients we've treated so far. What we saw in Pisces 2, the 2A study, were clinically meaningful improvements, most especially on the primary endpoint that we're going to be using in the current phase 2B study, and that is modified Rankin. And this slide really just demonstrates what modified Rankin as a measure is about. It's a measure of dependence in disabled patients, a 6-point scale, and as you can see from the chart here, the lower you are on that measure, the less disabled or the less dependent you are. The graph actually is taken from a registry study done in disabled stroke patients in Sweden a few years back in over 40,000 patients, and shows actually pharmacoeconomically that the best bang for your buck, if you will, comes if you can take patients down from modified Rankin 4 to 3 or 3 to 2. And indeed, those are the entry criteria, or one of the entry criteria for the phase 2B study that we're currently embarked on in the US. Where we're looking to take patients down at least one measure on this scale with a start point of MRS 4 or 3. So the pharmacoeconomic benefits and rationale for this treatment, if we can achieve that, are very substantial indeed. And that provides the motivation for us to move forward into a phase 2b study, again, based on a very meaningful response rate against modified Rankin that we saw in the phase 2a study. So Pisces 3, the phase 2b study, is 110 patients randomized against placebo surgery, which is as close as you're going to get to a placebo control in this type of intervention. Modified Rankin is the primary endpoints. There are a number of secondaries, as you see there. Treating patients at least 6 months after their stroke with modified Rankin 3 or 4 as the entry level. And importantly, some residual arm movement. What we learned from the phase 2a study was that if patients presented with really no ability to move the arm on the affected side, then they were very unlikely to be a responder to this intervention. So we are eliminating patients that have no residual arm movement in this study, in the phase 2b study. Okay, and finally, just a quick word on the exosome platform. So, as I'm sure you know, exosomes are nanoparticles secreted from cells. In the past, they are associated with the conveyance of cellular junk, if you like, but are now known to play a far more important role in cell-to-cell communication. And what ReNeuron is trying to do, in common with a number of other players that are exploiting the exosomic potential of their own agents, is to look at exosomes potentially as a novel delivery vehicle. And importantly for us, because we're harvesting exosomes from our own CTX cell line, which is an immortalized line, it does ensure a consistent exosome product time and time again. So it's very easy for us to harvest well-characterized exosomes, essentially from the spent media from GMP CTX cell manufacture or cell expansion. We've also been able to show an ability to load exosomes with small RNAs and proteins of various types. We've put a pretty extensive IP portfolio around this. And indeed, we can actually modify the exosomes themselves as well to carry other agents of interest targeting particular tissue types. And this is something that we're going to be doing collaboratively with a number of players that are looking for a novel delivery system for their own agents. We've signed one such deal at the moment thus far with a US business, and we're looking to sign a number of others over the near to medium term. And with that, unless there are questions, I'll leave it there. Thank you very much.